Okay, Bio30s, and welcome to our discussion today about sex-linked traits and multiple and lethal alleles. To start our conversation today, basically all the genes and all the alleles we've been discussing thus far in this classical genetics unit, they're all basically, the presumption is that they're all found on autosomes, which in a human, which would be chromosome pair 1 to 22. There are some genes, obviously, that are found on the sex chromosomes, on chromosome pair 23, whether that be an XX chromosome configuration for a female or an XY configuration for a male. So today I'd like to discuss a little bit about that and how, if those genes are found on, well, I guess with the genes that are found on the sex chromosomes, the ramifications that that's going to have on the transmission of those traits to the next generation, specifically as it applies to gender. So, for starters, sex linkage, again, is any case where in most cases, sex linkage will be a gene that's found on the X chromosome, but occasionally it will be found on, you can find some genes on a Y chromosome, which would be obviously specific to males only because females don't have a Y chromosome. You also might remember that the X and the Y chromosome are very different in size and shape. They are not homologous to each other. The X chromosome is much larger much larger than the Y chromosome, and that's going to have some implications as well. There are a few cases, as I mentioned. In most cases, sex linkage is on the X chromosome, but there are a few cases where you get a trait that's specific to the Y chromosome. An example of that would be, as this photograph shows, that isn't hair on the guy's head. That's actually hair on his earlobes. Growth of hair on the earlobes is specifically a Y trait. And I must tell you, I'll be honest, in my Bio30s, I have that gene because I do get hair growth on my earlobes. I can't grow hair on my head, but lucky me, I get to grow hair on my earlobes. So that is a wonderful, wonderful trait to have. But males are the only ones who have it because it is a trait, a gene, an allele that is only found on the Y chromosome, so therefore females can't get it. So what would the X-linked traits, how would this be, is there a pattern that we can see with the transmission of an X-linked gene? Well, there is. Here's one example. Fathers who have an X-linked gene will only pass that gene down to their daughters. They will not pass that gene down to their sons. And that makes sense because a male would have an XY chromosome pairing. The X chromosome, if a male gamete gets the X chromosome, then if it fertilizes an egg, then that offspring will become a female. So a male can only pass down an X-linked condition to his daughter. He cannot pass down that condition to his son because he's only passing a Y chromosome down to his son. Mothers can pass the sex-linked conditions down to both genders, to both sons and daughters. And in females, the sex-linked recessive traits can only be expressed if it's a recessive condition. A female requires 2 of the alleles in order to get the disorder. However, in a male, because a male has an XY chromosome configuration, a male requires only 1 of the recessive alleles in order to get that condition. So for all intents and purposes, a female is less likely to have an X-linked disorder if it's recessive than a male would, because a male requires only the 1 allele, which he will pretty much always get from his mother. So if if a male has an X-linked disorder, 100% that he's got he contracted that disease or that allele from his mother, because again, a son would only contract a Y chromosome from his father, not the X chromosome. So there are some unique inheritance patterns, and I'm going to be discussing that more as we go into the unit, into the last section of the unit, which will be my next video, which will be on pedigrees, where we'll be able to actually chart that in kind of a family tree scenario. But we will get back to that. Sex-linked traits works like they do because technically females only have 1 functioning X chromosome. That is something that we haven't really discussed. But females do have 2 X chromosomes, but one of them will form into something known as a bar body, which is a term that's used to describe the one X chromosome that isn't really a functional one. This inactivation is random, so in any cell, an inactive chromosome or a sex chromosome could either be from the mother or the father. But strictly speaking, though, a female with an X-linked disorder still requires 2 copies of the allele in order to get that disease themselves. And I'll give you a couple of examples here in a second. Well, in fact, I'll give them to you right now. A couple of examples of X-linked recessive disorders include the following. Color blindness, which we talked about earlier in the unit when we discussed sensory perception. I discussed to you red-green color blindness, which is the most common form of color blindness that is transmitted on the X chromosome. Duchenne muscular dystrophy, which is a form of muscular dystrophy that's a very particularly devastating form of muscular dystrophy. Hemophilia, which I'll get to here in a couple seconds. There's actually a form of rickets, which is called X-linked hypophosphatemia. I can say that really quickly, but yeah, that's a form of rickets, which is a disorder of the joints and the bones. Ocular albinism, so discoloration or a lack of pigment in the eye. And there's actually one called X-Men disease that's also found on only the X chromosome. Now, before I move on to lethal alleles, I wanted to kind of just show you how you actually show X-linked disorders. How would you write them as a genotype? So the example I'm going to use is going to be hemophilia, which I just mentioned is a recessive allele that's found on the X chromosome only. There would be 2 versions of the allele. There would be the normal dominant allele, which would not give you hemophilia, and the recessive allele, which would give you hemophilia. And just a reminder as to what hemophilia is, we covered it in Bio 20, but hemophilia is a disorder that affects the clotting of the blood. So if you have hemophilia, your blood doesn't clot properly, and if you have severe hemophilia, you can actually die from even a simple little bruise because there's no way for your body to clot the blood once the blood kind of starts flowing. So in this case, we have 2 different alleles. You'll notice that when I show a X-linked allele, it always starts off with an X. I have to indicate the chromosome. Now, technically, there would also be a Y chromosome in here as well, which just equals a male chromosome. But because it's an X-linked condition, the Y male chromosome is just going to be shown as a Y chromosome because this allele is not found on the Y chromosome. But when we're doing Punnett squares for X-linked conditions, we're not only just trying to figure out the ratios of the distribution of the alleles, but we also need to see the distribution of the males and the females. Because technically you would have a male. So let's say I had a male who is a normal male. A normal male would have this genotype. He would have a normal X chromosome and he would have a Y chromosome. So that would be what I would punch into my Punnett square. I would have X capital H Y. So I would have an X capital H Y on the top of my Punnett square here to represent the male in the pairing. Now, if I had a female that was hemophiliac, who he was, mating with, that female would have 2 X chromosomes, and because she's hemophiliac, she would have to have 2 copies of the recessive allele. So she would have X little h, so that would go X little h. Now I can run my Punnett square, and not only am I going to determine the outcome of all the offspring, but I'm also going to determine the gender of the offspring as well. So this half of my Punnett square, because this would have been X, H, Y, all of these offspring on this side are going to be males. All the ones on this side are going to be female. But I would also determine whether or not they have hemophilia or not. So that's how you draw the alleles if it's an X-linked condition or... Now, if it's a Y-linked condition, because we don't discuss a lot of Y-linked conditions, but let's say I had a condition that was on the Y chromosome, well, then I would have to put a notation, in this case, beside the Y chromosome. But because this is an X-linked condition, we're not going to put that designation there for now. But just so you know that designation could be put on so for example, the hair on my earlobes gene, the recessive gene for that would be Y, and in this case, it's hair, so maybe Y little h. If I didn't have that condition, I would have Y capital H because the dominant allele does not give you hair on your earlobes. But that would be how you would express it on the Y chromosome. Okay. So here's a couple of questions for you. I want you to run a couple of Punnett squares on your own. You're going to pause the video and then I'll give you the answers. I got a male hemophiliac who's going to actually mate with a female carrier of the disorder. So she's a carrier. She doesn't actually have the disease, but she is a carrier of the hemophilia gene. I need you to give me the genotypes of the parents first, because you obviously need to know the genotypes before you plug them into your Punnett square. And then once you plug them into your Punnett square, I would like you to calculate the genome and the phenotypes of the offspring. Pause me right now, because I'm about to give the answer. Pause me right now if you need to, because here comes the answer. Here it is. Did you pause me yet? Okay. Got to be a little bit funny sometimes. Not that funny. All right. Anyways, here's what's going to happen. I got male hemophiliac. All right. Male hemophiliac. I know he's got to be a male, so he's got to have a Y chromosome. So there's half of my genotype. I'm going to have an X chromosome for him as well. I know the female is going to be XX. This is always going to be the starting point for my genotypes. I got a male, XY, female, XX. It's a Xlinked disorder. He is a hemophiliac, so he's going to have the small h because he has the recessive trait. The female is a carrier, which is code for heterozygous. She carries the hemophilia gene, but she does not actually have hemophilia. If she had hemophilia, we would call her a female hemophiliac. If she was not a carrier, we would just say that she is hemophilia free, or she's hemophiliac allele free. So in this case, we're dealing with a heterozygous mix. So she's going to be capital X capital H, X little h. Now I'm going to plug all that information into my Punnett square. If you didn't get that part of the question right and you want to pause me right now to try to do the Punnett square over again, pause me right now before I give the answer. Here's the answer. All right, so I've got my Punnett square in here now. So I'm going to have one offspring that's going to be X capital H X little h. I've got X little h X little h. I've got X capital H X Y, pardon me, and then I've got X little h Y. So those are my genotypes. I'm going to have, these are the genotypes in the question. I've got X capital H, X little h, X little h, X little h, X capital H, Y, X little h, Y, and then, X capital H, Y would be my two males. So here's my male, female, female. What are the phenotypes going to be? This person is going to have the phenotype non-hemophiliac, although she is a carrier, but she's non-hemophiliac, so technically that's the phenotype. You can't say they're a carrier because you can't tell they're a carrier by looking at them. Phenotype is what you can visibly see on the outside. So they would be a non-hemophiliac. Male is a non-hemophiliac up here because he's got the capital X capital H. Female hemophiliac, male hemophiliac. So the offspring odds would be half the offspring would be hemophiliac, half the offspring would be non-hemophiliac. Now remember with these questions as well, questions could ask you the percentages of males versus females. In this case, the ratios between males and females are the same, but that isn't always going to be the case. In some cases, the ratio of my offspring and the disease found in a female set of the population of the offspring are going to be different than the male side. So be careful when you see the questions on an exam or a quiz or a worksheet, because it might ask you the percentages of all the offspring, or it could ask you the percentages of just one of the genders. So be careful when you read the questions to make sure you're actually answering the right percentages. You're either dealing with a 1 in 4 or a 4, either 0, 25, 50, 75, or 100 on all the offspring, or if it's just one gender, you're looking at either an answer of 0, 50, or 100, because there's only 2 of the offspring that are going to be male, 2 of the offspring that are going to be female. Hope that makes sense, but that's how sex-linked disorders are basically charted. And we are going to discuss a little bit more about that in the last section of the unit next time when I discuss pedigree. So there'll be more for this to come, and we will get back to hemophilia during that section as well. All right. Before we finish up today, a couple more concepts to discuss. First of all, we have what are called lethal alleles. Kind of an obvious name. Lethal allele means an allele that's going to kill you. Kind of an obvious one. These are certain disorders that are going to be lethal to offspring potentially before they can reproduce. An example of this, not always, but in a lot of times it'll happen before they can reproduce. Huntington's disease is an allele that pretty much kills you. If you get Huntington's, it's sort of a death sentence. There's also an allele found in a type of cat known as a Manx cat. If you like cats, you might have heard of a Manx cat. Manx cats are tailless cats that are bred. They originally came from the Isle of Man in the United Kingdom. These tailless cats are created because of an incomplete dominant set of alleles. You've got the allele called the ML allele, and then you've got the regular allele, which is the M allele. These control spine and tail growth. So if I've got an MM genotype for the cat, just regular alleles for cat or tail growth, I'm going to have a normal tail on the cat. If I have a heterozygous mix of the alleles of an M and an ML allele, I'm going to get my Manx cats, my tailless cats. However, if I have a homozygous configuration of ML-ML, that's going to basically be lethal to the embryo when it develops. There'll be severe spinal deformations of the embryo, and the embryo will essentially be stillborn. It will not survive. So these Manx cats, when they're bred, have a 50% chance of creating another Manx cat. They have a 25% chance of running a regular cat with a regular tail, and they have a 25% chance of producing an offspring that's going to die in the process. So that's sort of another example of a lethal allele. But in this case, the lethal allele only happens if it is in a homozygous configuration. It's not lethal if it's in a heterozygous configuration. It just creates, in this case, a tailless Manx cat. So that's another example. Okay, one more, multiple alleles, and this is the last big concept of today. Multiple alleles refer to, again, when an organism has more than one allele that controls a specific trait. We kind of talked about this with blood typing. We had our I, A, I, B, and then little I, 3 alleles, but that was a little bit of a weird one because we had 2 that were codominant to each other, and then we had the little I, which was recessive to the other 2. Generally speaking, with multiple alleles, you could have more than 3, and usually when you see questions like this, they're going to give you, generally speaking in this course, there's going to be 4 alleles. Now, there could be more, there could be 5 or 6, but generally speaking, most of the questions you see for multiple alleles, what I've seen them for Bio30 are usually they're dealing with 4 alleles. And it is an order of dominance. So, for example, on the page here, we're dealing with, we're going back to our fruit flies. Thomas Hunt Morgan and his fruit fly experiments, they discovered there were certain alleles that were, you would have multiple phenotypes. So you could get different colors of fruit fly eyes. There was red coloration, apricot, honey, and white coloration to the eyes. And based on how the combinations of these alleles came together, you would get any one of these 4 types of phenotypes that would originate. But they discovered there were 4 alleles that controlled eye color. And because it's an order of dominance or multiple allele, we are using a number notation in this question. Now, anytime you get a question about multiple alleles, there will be a very strict setup in the question. They will tell you, here are the names of the alleles, and sometimes you'll see small letters and large letters, and there's all sorts of configurations that can be used for multiple allele questions. There's no set fast rule to what the alleles are going to look like, but in the questions, they're going to set up what the allele designations are and what the order of dominance of the allele is. And if they don't do that, it's kind of impossible to answer the question if you're not given the order of dominance, and they will generally give you what the allele configurations or what the allele designations are. In this case, in this question, I've used E1, E2, E3, E4 in this case. This makes it a little easier for you. E1 is the most dominant allele. E4 is the least dominant allele. E1 would be dominant to E2, 3, and 4. E2 would be dominant to E3 and E4, but not to E1. E3 would be dominant to E4, but not to E2 and E1. And E4 would not be dominant to any of the other 3 alleles. That organism with the white eyes would have to have a homozygous configuration. They would have to have an E4, E4 in order to basically, in order to create, E4, E4 would give them white eyes. You would have no other configurations possible. If E4 was with any of the other alleles, it's going to basically just get shut down. It's not going to happen at all. I am going to do one thing here because I kind of did this question a little bit backward. I'm just going to do this. to change those numbers around a little bit. I'm not quite happy with what I did there, but I'll get to that in a second. So here's what I'd like you to do. On the bottom of the page, or on the bottom of my board here, I've got an E4E1 is mating with an E4E3, and then I've got an E3E2 is mating with an E3E1. Those are the genotypes of the parents. Here's what I'd like you to do. I want you to tell me the phenotypes of these parents. What would the eye color be of each of the parents? And then once you've done that, I would like you to then calculate what the phenotypes of the offspring of each of these parents would be. Pause me right now because I'm about to go over the answer. Here we go. I got an E4 and E1 and E4 and E3. Well, I've got 1 and 3 are the dominant alleles there. This organism here is going to be red-eyed because there is one E1 allele there. It's the most dominant allele between the two. So it's going to be red eyes. E4, E3, 3 is dominant to 4 in this case. So this one's going to be honey. So I'm going to have a red and a honey for the first pairing. If I take those and put them into my Punnett square, I got E4, E1. I got E4, E1, or E4, E3, pardon me. So I'm going to have an E4, E4 here. This one's going to have white eyes. E4, E3 is going to give me honey because honey is the dominant allele there. I've got an E4, E1, so that's going to be red eyes. And then I got an E1, E3, that's also going to be red eyes. So 50% of my offspring are going to have red eyes, 25% of my offspring will have white eyes, and 25% of my offspring will have honey eyes. That's for the first pairing. All right, the second one is going to be, I've got E3, E2, and I've got E3, E3, E2, and E3, E1. Let me just draw them into my Punnett square first, so I've got those in place. And I can do all my numbers later on. Okay, E3, E2. E2 is the dominant allele there. It's dominant to 3. So this one's going to be an apricot-eyed fly. And then the E3, E1 is going to be red-eyed because E1 is the dominant allele there. So I'm going to have a apricot and a red-eyed fly having some offspring. So let's see what the offspring are going to be. I got E3, E3. That's going to be honey because it's both the honey allele. I got E3, E1. E1 is going to be dominant there, so that's going to be red eyes there. I got E2, E3. E2 is dominant to E3, so that's going to be my apricot-eyed offspring. And then I've got E1, E2. E1 is dominant to E2, so it's going to be red-eyed. So in this case, 50% of my offspring are going to have red eyes, 25% of my offspring are going to have honey eyes, and 25% of my offspring are going to have apricot eyes. There you go. That's multiple alleles. Actually, not that difficult. It's just the whole thing. There's a couple more here on the slide, right here. See if you can run these through really quickly. I got E1, E4, I got E1, E4. Oh, sorry, no, I already covered that. Yeah. This is just showing you what the phenotypes are. I should really read my notes because I should just, rather than putting my foot in my mouth, read that. This was way better than what that little bullet said. All right, let's finish it up. Last thing for today, polygenic traits is pretty straightforward. Polygenic trait just means that you got a trait that's controlled by more than one gene, potentially on more than one chromosome. These would be what we call continuous traits. We talked about these. This is going back to grade 9. There were continuous versus discrete traits. Either a discrete trait would be like either I have it or I don't have it. Hitchhiker's thumb versus non-hitchhiker's thumb, like you either have the trait or you don't. Continuous traits would be things like eye color. There's all sorts of variations of eye color, height, skin color. These are all continuous traits. And generally speaking, these are controlled by multiple alleles, so they are also considered to be polygenic traits as well. That's what a polygenic trait is. And finally, let's do a quick recap of Mendel's laws of inheritance before we move on to pedigrees next day. I discussed one of them specifically, which was the law of segregation. But there's also the law of independent assortment. You understand what independent assortment is. The lining up of the chromosomes at cell division, whether they line up like this or like this, it's like a flip of a coin. So that rule as well. And then there's also the law of dominance, which he discussed that one allele could be dominant to another. Again, I had mentioned that Mendel was probably a little lucky when he chose his pea plants to do his experiments on, because if he had chosen some other plants that have, let's say, co-dominant or incomplete dominant traits, he may not have gotten the same results. So, for example, law of segregation, he said that the inherited traits are based on at least two factors, which we call genes or alleles, and they behave as discrete units. These genes separate randomly from each other in the formation of gametes, and then each gamete receives one copy of the gene. Had he chosen an organism that didn't have complete dominance, he may not have seen that. He would have seen a blending of traits. A purple and a white flower, for example, would have given him a lilac flower rather than a purple and a white giving him a purple flower, because of the complete dominant alleles that were involved in coloration in the pea plant. So we got lucky there. How about independent assortment? Two alleles from one gene assort themselves independently for the alleles in relation to the alleles of other genes during gamete formation. Again, if you would have had codominant or incomplete dominant organisms, he may not have been able to come up with that conclusion. And finally, the law of dominance would have gone out the window, because he would have seen a blending of traits, not one trait dominating over another. So again, like I mentioned, Mendel, which was the start to all of these, the last 5 videos I've shown you, it was him who started this whole thing rolling. He did get rather lucky when he chose his pea plants over others. In fact, he chose another plant later on his research. I can't remember what the name of the plant was, but it was a codominant plant or incomplete dominant trait, and he did not get the results that he expected off of that plant. So, he really did choose wisely, or very luckily, when he chose his pea plants off the get-go. That's my discussion today on Xlinked traits and on lethal alleles and multiple alleles. Your homework is fairly well labeled. It is a question booklet entitled Sex-Linked Traits and Multiple Alleles. Go figure. They'll be due for next day. Next time, I'm going to finish up our classical genetics unit with a discussion on pedigrees, how we use family pedigrees to chart genetic history, and also to determine what type of inheritance is being discovered from generation to generation, how autosomal traits will inherit differently through generations, say compared to traits that are carried down by the X chromosome or the Y chromosome. So won't you join me next time for that discussion, and that will finish up the unit. Until then, thanks for tuning in, and we'll see you next time.